Can you hear me okay?
Yep.
Yeah, I have a little bit issue with my headphone, so hopefully if I didn't hear you well, um just maybe like do a reaction and I'll be able to hear you better. So maybe before we move into that, let me check the slide sharing real quick. Let's see. Dun, 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 dun. Mhm. Right. Can someone let me know if you can see my slide?
Yeah. Yep.
Cool. And you can hear me okay, right?
Yep. Loud and clear.
Sounds good. All right. So, um, I hope everyone is has been learning a lot about the basic of the synthetic biology system so far. Today is kind of like one of the very important system because most of the time when we work on biology, we have to read them and be able to interpret them properly. So this is a quick session um, of the process in which we're gonna try to read the languages that was provided in the nature. So these are very, interesting way of us trying to go through how the DNA can be read and interpret and translated into other kind of system. So it's supposed to be around 40 minutes of me talking and sharing, but it's also going to be a an overview showing the program and how to use it effectively to you guys as well. So there will be more of these content in the coursework. So if you don't understand everything in these, in intro talks, it's fine, and you see that again. So let's get started. Um, so the process of the DNA and the other, overview, we're gonna be talking not mainly about how do we use a specific tools so that we can read these DNA language properly. So similar to like how we try to read, other languages, our writing system is usually, you know, um, readable when we try to open it on some program, simple program like Notepad, Microsoft Word, or Google Docs, something like that. The reason I, show these three, system is because like there are different complexity in our system when we are trying to read different kind of data, right? And in the further demo today, I'm going to show you some kind of process where we try to attach primers, which is the short DNA part, onto the program called Benchling as well, and see if we can try to make the in silico PCF fragments from these, and also trying to navigate some kind of simple assembly wizard on Benchling as well. Um, and together we're going to try to use, some examples from the first and protein work that we have been, discussing on Monday to see if we can, show some different scenarios of the molecular cloning, and we get into that later on. So first, I really like this slide that I have been using to talk with everyone everywhere. That DNA most of the time they will be stored as a key biological information. So you have DNA, I have DNA. They are slightly different. Bacteria also have DNA, and most of the time, these DNA will be the key form of information that was used to store it and then like getting read and also translate into RNA and protein so they can function accordingly. And to be specific about the biotechnology processes in the past 20 or 40 years, most of the DNA that we are working on are usually passed through the form of plasmid DNA, in which it's a circular DNA that we use to engineer. And for this plasmid DNA to visualize them even further, you can sort of like segment them into a Lego pieces. What you see here is, one of the, the key paper in the field where we try to assemble different pieces of DNA together. It's just like a Lego. What you can see right here, maybe like you can read the type 1, 2, 3, 4. These are the pieces that can be connected using the molecular assembly process so that we can form a single plasmid that has a lot of these coding sequence um, or programmed DNA together. So again, without our knowledge, it would just look like a gibberish, DNA information. But with our process of annotation and also like analyzing, we're going to be able to annotate this part according to their function. So in the plasmid, if you are working on the plasmid that can work in bacteria most of the time, they will need three main DNA context or elements that will be needed. Otherwise, it's not going to be a plasmid that can function properly. Those three things are the origin of replication for the DNA replication. DNA can, duplicate themselves. They will just use its own sequence to make another piece of DNA over and over again. This process is called replication. And the origin of replication is the most important thing on the plasmid. Otherwise, they cannot make another copy of themselves. The other thing that is very important is the selectable marker, because when we're trying to put plasmid onto any organism, we want to make sure that the organism that we take out has the plasmid. So it can be any other thing. It can be like, some kind of antibiotic resistance. It could be like metabolic genes, but it needs to be in a property where we can select them. And the third one is technically not needed, but if you don't have it, it's kind of like not as useful, in which these are the gene of interest you're trying to put into these new parts of DNA. So these are the three key elements when you are trying to engineer anything. Most of the time, the origin and the selectable marker are not necessarily having to change, but the gene of interest is always like something that we will keep changing over and over so that they have different properties, like having different colors, for example. So we'll see that in the later system. The reason that we use plasmid as a DNA to work around these processes is because it's very easy to put plasmid into bacteria. So these are some schematics in which we can show some kind of like, heat shock or electricity shock so that we can make some pores in the bacterial surface so that these kind of, small DNA can get into the cells. So what you can see right here is that like similar to trying to break glasses, we just like make it put the back microbe into the cold temperature and then heat it up so that there's some expansion, make some pores, and the DNA can get in, and then we just cool it down again to trap the DNA inside. Similar thing with the electricity, you just sort of like shock it so that the membrane sort of like make some pores, they're getting, taking some of these DNA inside, and then we're gonna trying to grow them further so that they can kind of repair themselves, and some of the microbes that has this DNA inside will be able to survive based on the selection that we put onto the plate. Um, we also have another system in which we can use, one bacteria to donate another DNA to the other one, and that was called conjugation, where they form this kind of microstructure where they can transfer this DNA from one organism to the other. That's also like, another interesting process to work on. Um, but in most of the class, we're focusing on technically the the transformation process, which is a lot more simple using the heat process. So um, when we talk about this DNA, we can draw, a schematic or a figure to represent what they are. But technically, they're actually the feature, the kind of, property that they have is based on just a few simple characters that we can represent them in different format. So what I'm trying to show you here is the DNA that represents the genome of E. coli. So they have this kind of code saying like, U00096. These are the code of, bacterial DNA, in which these DNA will just get translated into RNA and protein and show the function later on. This format is called FASTA or FASTA. The way that it was organized is that like it's gonna have these, the symbol, more right here, and also having the name of these sequences and just a string, just a list of characters ATGC, which represents the DNA. So what does it mean by ATGC is these small part of the DNA that was connected. We will call the 5 prime as like the left-hand side of the DNA, and the 3 prime in as the other part of the DNA. The DNA is composed of three different things. You may have heard about that from the previous talk already. But the key things that were different of the DNA is this nucleobase, and this part, the green thing, and the other color part represent different DNA moiety. So these things we refer to A, T, G, or C in this coding sequence if we're trying to read into. Um, so we sort of like, simplified them in these simple, um, Roman characters, or Latin characters that we've been using so far. One thing that I want to emphasize is that, most of the time the DNA is in the double helix form, and they actually have the other strand as well. Even though we have, this kind of set of characters that show us what the base are oriented, organized from left to right, it doesn't really capture the actual DNA structure because DNA will have another strand that was complementing. And because it's not just a copycat, it's a complementation. A will bind with T and C will bind with G. If you have, a proper way of reading it, you should be able to encapsulate the other strand information as well. So that's why, this kind of like Microsoft Word or Notepad is not the best tools because it will give you only one strand of the DNA. Um, if you're trying to look for some kind of DNA online to see that, how can you download and visualize them, you can go into this website in CBI, where is the one that was provided by the US NIH. And you have a lot of database of information right there. You may have heard about this from our team already as well. But this process, this website will show you the GenBank format of this DNA. And it's very similar to like what you've seen when you open it in the Google Doc or the Microsoft Words. So because we have these, it doesn't mean that, it's the best tool to use so far because like when you see this DNA, if you try to open it in the proper tools, like the program called SnapGene or the cloud system on Benchling, it's gonna help you visualize the other strand. Just this is very important because when you're trying to search for a certain DNA sequence, it could be either on the top strand or the bottom strand. So by populating these two-stranded nature of the DNA is a very important features of these tools where you don't have to store information twice, right? It's similar to like when you're reading something from, the folder. If you know that, the other strand will be duplicated properly, you don't have to store that as a file, but it's going to be generated on the computer to show you what the other parts are. And when you're trying to search for, let's say, ATGZ sequence, you're going to be able to search in this kind of format that we will also find from the top strand and also the bottom strand. We'll see that when we go into the demo quickly. So another thing that was very important before we go into the web tools is that this information were derived from the chemical processes where we read the characters of those part that we call nucleobase. So let's say we label them with different colors and try to analyze them. This is the way of the DNA sequencing in which you can generate a kind of plot in which each color of this peak will represent one DNA. So this DNA sequencing came from the process called Sanger sequencing, and the Sanger sequencing is the process which received the Nobel Prize like from 30 or 40 years ago already, and it has been used by a lot. But we also have the other generation where it can give us a lot more comprehensive data. Rather than just using the Sanger sequencing. So, you might be interested into looking into sequencing processes further, but this is like in a snapshot. When you try to open this information in the DNA reading process, you'll be able to compare whether the actual DNA that you have match the DNA that you are trying to make or not. So that's a very important process. When we make any DNA in the lab, the important thing that we're trying to make sure that is working is that they match what we are trying to get. If there is any missing information, missing base, it can just like turn our DNA into something that's not usable anymore. So those could be called something like mutations or some deletion of process of this DNA. Okay, so if you were to use any DNA tools, I would suggest to use, Benchling rather than the SnapGene process because the SnapGene application, there are two versions of them. There was a viewer in which is free, where you can take a look at any DNA. The graphics is also pretty good, but they don't have the full feature in which you have to pay for the full price for getting the actual SnapGene rather than the viewer version. Otherwise, you're not going to have the overall process. So, you know, for Benchling, if you register with the email that was associated with the academic institution, you're gonna get that for free. So that's a very good process. So that you can sort of like try to use Benchling, and that's the tools that I'm going to show you today on the simple forms of the DNA analysis process. All right. So what we're gonna show you today is we're gonna go to the Benchling project, and the slide that I'm sharing to the BioBootCamp website should already have these project on the Benchling. So if you go to the slide and just click onto this one, it should have lead you to this kind of Benchling folder. Let's see. So can some of us, say it out loud or confirm with me on the chat that whether you can see the browser right now?
Just fine. Yes.
Perfect. Yeah. So on this browser is the process in which we can look into
Sorry.
what is available
No,
in
we
terms of
don't
We,
see
DNA.
it.
We don't see it.
We
yeah.
don't see it. We see
Can
your
you see anything?
slide deck, Ice.
Is it still the slide deck?
Yep.
Yes.
Not
We
the
cannot
not
share
the
the whole
not the
screen. We
PowerPoint?
need to,
Okay.
you know.
Sorry, not the actual thing. Give me 1 second.
And maybe you can put the link in the chat.
Yes.
Maybe to unshare and then share again.
Yeah. Let me try to do that.
Cool.
Yeah, the link on the chat is the correct one. You can see that from the PowerPoint that we shared previously. Thank you for putting that up. Let's see. Yeah, I'm not entirely sure if this Zoom allowed me to share the whole screen. Um, let's see. Let's see if we can do it like this. So now, do you see um
Yes, we
the
do.
browser?
Yeah.
Okay, perfect. Yeah, so briefly, what I want to show you in this browser, there are thorough information right here, but let's get into this arbitrary, new information here. So let's say there's nothing right here. I want to make a new piece of DNA. Just click this plus button, and you can make new DNA amino acid sequence or oligo or whatever process right here. There's a lot that they were providing, but let's go for new DNA or RNA sequence. So I'm gonna type it down like new DNA. And you can also tell them whether it is going to be a linear or circular DNA. So let's assume that it's a linear first. Just create. It's going to give you something similar to the blank file or the blank information that you may have when you open a notepad or a Google Doc or a Microsoft Word. So you type down, you may not see my keyboard right now, but I'm gonna type A. What you can see right here is that we actually add, these amino acids, like this nucleic acid sequence, the DNA sequence into this file, and we add more things. Let's say, I'm just gonna try to type gibberish thing, and you see that like sometime they're gonna show some kind of error because these are the sequence that is not matching with the DNA sequence. You will see that there's something apart from the HTGAA that can be written down. Those are actually, a group of DNA. For example, if I type down N, these N represent like any base. So for example, if I'm put it here and I'm trying to search for A, they would tell me that, in this sequence, there's like the sequence AAAA right there. To pop this thing up is just using Control F, the regular function that you will be using elsewhere in other word editor. But for these kind of like, other characters, if you type AA as well, it's not going to show up. But you type NN, that thing is going to show up. So this is, some example of, how to use these processes, because sometimes you want to look for some kind of DNA that were 6 DNA, but have, any other arbitrary base afterward. If it's something that, N at AAA, and you just add another N right there, it's not going to find anything. So this is sort of like, a simple process in which you can, take it and try to work around it. One important function that I want to show you here is the import process of the DNA. So when I click import, instead of, this is a create new, this is what we see previously, this is upload, and there's also import from the database. In this process, you can go to, some other links outside, and just try to look for certain DNA so that you can take that DNA and try to upload into this process. So let me try a quick, download. Go to Google. I'm going to look for maybe PUC19 plasmid, and I'm going to say maybe NCBI would be where I want to take it from. So when I go into this NCBI, this is the GenBank name. We usually call it accession code. And what you can see right here is actually the GenBank format. If you download this file and open in the Microsoft Word, you're going to see this kind of form information. But you take just this code and go back to the import process and type it down right here like this. Try to search for it. You see this, synthetic PUC19 vector name popping up. If you hit import, you're going to get this DNA sequence in a plasmid form. So it also tells that this, DNA is a circular DNA. You have this kind of like this source that was labeled for something that, came from other source. You can change the annotation. You can edit by changing the name or by deleting it. Most of the time, when I get it from the source, I usually get rid of the source because it's the whole thing that we've got. You would try to find, ATGC sequence. That's something that's popping up. Like specific sequence, if it was a promoter or the lac operon, for example, this one, it was labeled as a lac operon, and it just like copy and also try to search it. It's gonna highlight the part that is the lac operon information. You do try to paste this information elsewhere. Let's say pop it right here. It's gonna be just a string of ATGC threading together. Again, it's very important that we have the annotation process, right? Because like if you were asked to look at this sequence and try to interpret them, whether like what it is, it's going to be pretty difficult for us, for human eyes, to distinguish what is the key difference of these information. So having a computer to help us match the information, that's going to be pretty important and convenient. One thing that I want to know is that, if you try to use the search function in Benchling and you're missing one base, I'll try to delete one base from this thing. It's actually not showing anything anymore. So it's very important that the DNA, that you're trying to search for, is matching well with what you are trying to provide. So you see that I sort of delete one base right there. If I try to do the alignment process, sort of like to compare this sequence with the sequence that we have right here, I can do the alignment process where I click on the right and create new alignment. I'm just gonna paste the base right here and I'm gonna call it like one base deletion. So by doing that and go for the alignment, just use the default setup. What you can see right here is that this sequence is almost identical to what we have, except it has one base deletion right here. And this is the information that you want to see when you look at the DNA. When you try to build or synthesize a new thing, you really want to make sure that they're correct. And this kind of like one base deletion sometimes doesn't make any difference, sometimes make a big difference. It depending on where it is. It is at the promoter, it may just interrupt the promoter completely. If it's at the coding sequence, it may shift the frame completely so that your protein that you're trying to make is not the protein you want anymore. All right. So, you know, hopefully that give you some simple information. I see a lot of questions pop up. I'll try to go through the presentation first, and if you have time, you come back to the discussion later. Let's go back to the slide, and if you can tell me whether you can see the slide pop up instead of the browser. Can someone help me confirm if you can see
Yeah, confirmed.
you can see the slide now, right? All right,
Yes.
let's move forward. So this is another thing that we are trying to provide as a practice. So these are the phage T7 that we show in the day one of our presentation. So in this phage, the way that they were encoding this structure is by this, DNA genome, this phage lambda. If you try to, take the whole DNA out and try to use some enzyme to cut at different spots of the genome, you can make this kind of pattern. So I can try to show you how to do this exactly. But I would encourage everyone to try and get into Benchling and try to make this kind of digestion or ladder as well. So let's go back to the Benchling real quick. If you have, try to go to Google and try to look for the phage lambda DNA, you can do the same kind of import process, just like what I did. And you're going to get these kind of linear DNA of LAMCG, is actually lambda phage genome. If you try to go to this digest button on the right hand side, this digest that has like this scissor as an icon is actually showing us different enzyme that we can try to bind or cut this DNA part. So if we try Hindi3, which can cut 6 spot and just try to run digest, it's going to tell us that you're going to have these kind of different cuts, and you have, a DNA of different length from 23K to 2K to 9K. And Benchling can also give you a virtual digest where it gives you this kind of, pattern that you've got. So if we go back to the PowerPoint, it's going to match pretty much, what you can see from this slide if you use the same enzyme. These are also, the same of what was shown in the NEB site. Let's do a little bit of, practice of trying to do the edits. It's going to be a big shift of the thing we are trying to show here. Let's just assume that we have this DNA, that has CoE1 as an original propagation, has ampicillin resistant gene as a selection marker, and also the GFP as a coding sequence to make the bacteria capable of making the green fluorescent protein, as an example. So what we have here is that if you click into the PowerPoint link here, you see these parts together. And what we could try to do is that we're going to try to make these different parts together. When we make these different parts with these kind of, enzyme that can cut at different sites and then can match later on, specifically, we can actually connect these three pieces, like thread them together, like we connect the Lego, and then we can try to make this whole plasmid again from maybe like the DNA synthesis that we can also order from the vendor these days. So let's jump to the project. All right. So what we have right here, we're going to go to number 1, ligation folder. And this ligation folder, we have different parts available. So I sort of do some earlier work around already, trying to make these, part A, part A, B, and C. So you see them labeled as like 1A, 1B, and 1C. These 3 parts are something that's going to be connected to make a new ligation product. You can also see, some other ligation. There was like a dry run that I was doing by myself, but I'm going to show you live here to see how it can be used to assemble. So if you go to the new assembly right here and use digest and ligate, start, you're able to, set some fragments right here to do the actual ligation. So for example, if you go to the CoE1 part at the backbone and trying to tell them, hey, we're going to use this as a backbone, and we're gonna cut at this site called BOX1, box one, and this other site called SGS1. Set from selection. Then you're gonna have this guideline that tell you how can they connect with the other parts. So what you can see right here on the left hand side, we call this a blunt end, and on the right hand side we call it sticky end, where there will be other bases right here that should match with the other sequence. Otherwise, they won't be able to connect seamlessly. So we go to part A, in which they can connect with this part A right here. You can see that SGS1 was also highlighted because it's the enzyme that was used to cut the other part. If I click right here on the SGS1 and try to click on the other part, in which I'm going to use, I think I'm use KFL1 and set from selection, you will see that they tell us that the SGS1 site sort of connects seamlessly right here. So when they do the connection, it's going to connect properly. But on the right-hand side, it can't really connect properly yet because the sticky end right here doesn't match with the blunt end on the right-hand side. So the reason that this happened is because like we plan to do a 3-part ligation. So if we add another insert right here and go to the part B and try to set this KFL1, and also the site called PSHA1 or box one right here and set from selection, you're gonna have this, proof of concept telling you that, hey, seems like all the connection looks okay. Looks like everything checks out. If you click done, they're gonna ask you what you want to name this one. Let's call it ligation 2026 and assemble them. They have like some notes telling that you're using blunt end, right? That's some kind of process that sometimes the process these days doesn't prefer the blunt end ligation anymore because it has less specificity to the other things. We try to compare this ligation to other sequence. Let's say 1_CoE1. That's the sequence that we can probably compare to. And you can see that this is exactly something that you want to make or not through this, DNA alignment process. Looks like it's something that you want. And this part has some kind of like a little bit of a annotation right here that was disconnected because it was a part that was cut from 2 different things. Along the same line, if you have this part somewhere else as a plasmid, and you sort of like want to make a fragment from the process that we call PCR, you can also, add these primers. So I have sort of like add some of these primers already, but the way you can add primers is by right-clicking, create primers. It's gonna have these information down here, and you can just tell them whether like these are the same primers that have been used, have been named elsewhere or not, because Benchling has access to all the primers that have been generated so far. So this short DNA sequence is going to be annotated as something called KFL1 forward, and it's called F, it's forward. You try to see if you can do primers, 2 primers that can connect these 2 parts together. I'm going to go down to maybe this position. If you click at 2 primers like this and go to this icon on the right hand side, you can try to pair them together. And if you link these 2 primers together, you can technically generate a PCR product by scrolling down right here. And it's going to give you a new sequence. So in this process, you should have this new sequence that can, link you to, the new way of making, a PCR products in silico. So far, hopefully, like, you can see this simple process of making the new plasmid and give you, a different information. All right. So let's get back to the slide. So again, like all the links were available through the PowerPoint. If you, want to redo it by yourself, you can feel free to go through the PowerPoint and just take a look at it. Maybe, what I'm trying to show you here is that we're trying to make the DNA that can express this screen fluorescent protein. And this screen fluorescent protein, when you put into the bacteria like E. coli, it's going to change its color from this kind of white bacteria into the green bacteria like this. So this is another kind of selection process where you have the function as a color. And in our process, we also like, having other kind of system. If you go to the classical blue-white selection process, you can see that they have this kind of picture um, in the internet by the classical cloning, where they use this blue-white screening instead based on the laxi gene. So I'm not going to go too far, but this is a previous approach where people check whether they have the laxi gene or not. And the new approach that people tends to do these days is by using the fluorescent protein instead because it's a lot easier and a lot more colorful. This is when you grow up in the liquid media, and you can see that, this is something that was like having GFP, and this one doesn't. And when you spin them down, when you sort of like trying to rotate them, or spin them at a very high frequency, they would just come down because they're kind of heavy. And at the sedimentation right here, you can see the color from the gene that we encoded in the gene of interest right here. So again, you can also do the same process by changing the GFP to another fluorescent protein like RFP right here. And if you change that GFP into RFP, what's going to happen? I mean, I'm not going to go to the benchling process anymore because I think like you've seen almost the same thing already. But please give it a try if you want to try to do this kind of like conversion from GFP to RFP. I'm going to show you that like, if you do this kind of PCR, you're going to generate this kind of PCR band where you can run it on the agarose gel. And this agarose gel can tell you what is the size of this thing. Um, and then you do the heat shock, and then you're gonna go through the kind of selection, and you're gonna have these, change of color from green to red. The real life scenario is that you're gonna have not just green, but also not just red, but also the green one as well, because there might be some background of the one that hasn't get converted from the green to the red one. But these are the process that would be a good takeaway for everyone to, use these kind of tools to read DNA. On the slide, there are more things where you can do, Guyanese design or, trying to use the Python to make the Golden Gate system. But I'm skipping all of that. If you're interested, you can, take a look further into the recording from the last year. But we're trying to make it shorter this year so that we can have more time for the Q&A and also like for other operation that we may want to talk on. So these are the key takeaway. I want to make sure that, after listening to this chat, you recognize that we can use these two like Benchling or SnapGene to compare the DNA, RNA, protein information. You can read, you can compare, you can edit, or you can even design them from scratch. Benchling is, the one that I would suggest rather than other ones because it's free. And these in silico operation may take some time for you to go through, but I think it's worth it because if you go through this, this in silico process, you'll be able to save a lot of the on lab time, the experiment time you're working inside the lab space. So if any of you were doing this experiment in the near future at the notes or anywhere at MIT, I would suggest to take more time to do the in silico or the on computer designs so that you can save a lot of time on the bench in which you're going to take like multiple days to do anything, right? So these are the resources that I put it on the slide so that you can look further on like, how things can be worked on. Spatial emphasis on this tool called Planetate, where you can actually put the DNA sequence onto the website and it's gonna pop up, the annotation of what DNA is. Let me just show you briefly then before we go to the Q&A. If I take, copy, Control C, copy this whole DNA sequence and go to this Planetate and give them the DNA sequence, this is something that you're going to get by the analysis of, this Planetate too. So it's telling us that there is a beta lactamase gene, there is ampicillin resistant gene, and there is like origin of transfer. Oh, seems like it's not everything that we were looking for, but that's probably because I was, taking this from the PCR. So it doesn't know whether it's a linear or not linear. You have to tell them on this process, on this like input, whether it's a linear or not. But overall, I hope these processes give you some kind of guidance on how to read through these information that was generated by the DNA so far. So I'm gonna make sure I go back and stop the sharing if there's nothing more. And hopefully, if there's any question, I will try to address in this talk. But a lot of these kind of information were also available in the previous recording. So if there is any question, please feel free to reach out to TAs or um, you know other coordinators, because I think these are the process everyone has been going through almost every day when we work on this kind of process. So hope you have some takeaways and we're able to try to use these tools at some point.
Awesome. Thanks so much, Ice. Great
Yeah.
talk. All right. So if there's any more questions for Ice, please post them in the chat. Otherwise, I will make
Mhm.
Greg.
Yeah. So maybe while Greg is setting things up, I'm gonna try to scroll through some kind of question down there. Um, so um, so there's an immediate question coming out, if you compare if you compare wet lab to computational or in silico, what is the value? I think like both of them have values. Computational approach can accelerate a lot of process, but most of the time, you try to design something on the computer, and you need to eat it so that those medicine can function properly in your body. You have to make it full circle. So most of the time, we try to combine these modern research in both the computational and also the wet lab so that we can use them efficiently. The problem that we had with the wet lab only in the past is that like the scale of it may not scale as we want it to be, and the the computational process or the pipeline that we had can accelerate a lot of the design approach without spending days and days or maybe years in the wet lab to explore things. So that's the clean thing that we are trying to emphasize now. So um, you know um, there's some question about the access to the slide. You see the information about this slide again, and you should be able to access all of those through the slide. And if you can't catch any of the last page of the document or whatever, try to get into the slide, and those should be shared to you via email, or you can find it on our website. Again, if you can't find any of the information for today's or in the past few days lectures on the BioBootCamp, all of this should be available, to uh, all of you through the website. All right. I think I go through, several questions available in the chat. And I think there are some notes about whether you can access the Benchling if you're not part of the academic institution. I think you can still apply. And if you have any question, try to, you know, check in with us. If you don't have these, access, we can try to connect to Benchling so that you have like the basic access to all these things. But I think you don't have to be from academic to have some other basic usage. You can try to just like use the Gmail or like other Outlook to try to get a information to read. And maybe you have to look through other connections to gain um, the same kind of information that we can share with you. All right. Um, Seems like I cover everything. I hope that's useful.
Okay.
Awesome.
If
Thanks
there
so
is
much,
any
Ice.
question, please feel free to get into the what do we have again? We have our office hours, right, Ethan?
Yep.
So
Yep. Yep.
please feel free to stop by on that one as well.
Perfect.
Okay, terrific. Thanks, Ice. So I'll just jump right in then.
All right, take it away,
So
Greg.